Thank you very much, Jesse, Susan, others who helped to put this together and make this run so well and bring us all together, many of us who have been collaborating and hopefully many future collaborations, as I've started to see already gelling here today. So yes, I'll be talking about the Malaria Host Pathogen Interaction Center. This has also become known as the MAPIC. So you'll be hearing me talk several times about MAPIC. And you can read all about it on the web at the Malaria Host Pathogen Interaction Center website. We have two co-principal investigators. One of them is your own Jesse Kissinger, and also Alberto Moreno from Emory. The website is systemsbiology.emory.edu or mapic.emory.edu. We work with several Plasmodium parasites, particularly the most predominant human malaria parasites, Vivax and Falciparum. We've already learned about how diverse these are, very different in also some of their biology, immunogenicity, etc., pathogenicity as well. We also work with the monkey malaria parasites that are closely related to Vivax. And we're at the point in time right now in this decade with a lot of challenges, particularly because we have all the genome pieces now, over 5,000, 6,000 genes in the parasites are known. We also have the host parasites from the human host and from the non-human primate hosts. And we need to exploit this information to find out more about malaria and how the infection actually takes place and how the parasitism actually works. We also have a lot of technologies we didn't have before. All these different omics to study the RNA in depth, the proteome, the lipidome, you name it, host pathogen interactions in ways that we couldn't do before. And we hear now students frequently talking, even at lunch, about host pathogen interactions. Several years ago, we didn't hear students saying they're interested in host pathogen interactions, even when they come and interview for grad school programs. So clearly it's becoming known that these are important for a lot of diseases, and definitely so for malaria. And there's just so much that we don't understand, and we aim to understand what these interactions are all about. So what is systems biology? The project today is all about systems biology, aiming to let us understand more about host pathogen interactions. It involves the holistic study of an entire biological system, not a specific point. And it can be a whole, maybe I just got this here. Yeah, a set of biochemical pathways within a cell, the entire cell itself, an organ, a whole organism, or interaction of different organisms. For the MAPIC, we're collecting comprehensive data sets. And then we're also working with computational biologists to actually model the disease and the infections. And ultimately, we believe we can learn things about the malaria infections that we just haven't been able to from smaller bodies of data. And actually, we're also interested in co-infections and comorbidities, taking into account as well the cultural environment and backgrounds of the communities involved. So a lot of things to think about when you're thinking about systems biology. And the challenge is making sense of what's going to be unprecedented data coming. It's one thing if you're studying the transcriptome or the proteome or a lipidome independently, or two of these different omics. It's another thing if you're looking at all of these bodies of data together and then trying to make sense of the mathematics, putting it into code, and coming back to understand what the biology is all about. And that's our challenge. And so MAPIC does involve a lot of clinical sampling and data collection from non-human primate infections, a lot of use of these omic tools, a lot of bioinformatics and computational modeling, ultimately to understand the infection. We'll be carrying out a lot of science internally to understand these infections, but particularly as well to get data and tools out to the scientific community at large, because there's going to be more data coming from this project than we as a group can possibly analyze. And it's a big group. Not all full-time, but a number of us are part-timing on this and part-timing on other things, but we're a big team, and yet we can't manage the entire project, so we'll need a lot of new helpers and new collaborations, again, which I hope more can come from the Georgia team here. And it involves, again, our program directors, lots of different core leaders, lots of staff, lots of students. We have interactive cores. Remember, this is one big project, not multiple small projects making a whole, but a big interactive project with multiple cores, including malaria, immune profiling, functional genomics, proteomics, lipidomics, metabolomics, informatics, mathematical modeling, and computational biology, all working very closely together. And we're a Georgia team, and we got to really, capitalize on that. It's quite amazing that we've had this amount of expertise in the state at Emory, UGA, Georgia Tech, CDC, and that we also have a lot of international collaborations, that we can work with all these primate models. We can work as well with the mosquito vectors all here in Georgia. How did we come to be? There was a call by NIH for a contract proposal in 2011. And the contract required a systems biology focus. Applicants were required to generate, integrate, and share multiple large omic data sets on host pathogen interactions for persistent and recurrent infections. That could have been bacteria, viral, other parasites, or malaria. And we obviously had all the tools and elements of this, putting out a malaria proposal. We did so in June, and the project started in September 2012. We had our title of Systems Biology of Malaria as a Model for Host Pathogen Interactions. As I said, it's now known as the MAPIC. It's a large contract of $19.4 million. But as you see, with so many people and so many different things involved, that money gets stretched. So we are looking as well for collaborations and other kind of projects to build in new grants and new training opportunities for students to enlarge the potential of the project. We have a central unifying hypothesis that non-human primate host interactions with Plasmodium pathogens as model systems will provide insights into mechanisms as well as indicators for human malarial disease conditions. In other words, when we're studying non-human primates, we are going to learn about the human infections and vice versa. My life cycle rendition involves emphasis on Plasmodium vivax, but also to make the point that vivax and falciparum infections are different, as is Knowles eye infections, which we've heard today are now a big problem in Southeast Asia. The focus of MAPIC, we're interested in the entire life cycle. We'd like to grow with more emphasis on liver and mosquito and gambina sites, but to begin, the focus is on the blood stage of the disease, which already we've heard a lot about today. I'd like to bring some new emphasis to a few points about that part of the life cycle. In particular, merozoite. We've yet to hear too much today about the merozoite. To begin the process of infecting the red blood cell, merozoite attaches at any point on its surface. It must reorient, and then it attaches at the apical pole. It then burrows its way in a kind of zippering effect into the red blood cell, and then begins to metamorphose and take over the red cell as modifications like Dave mentioned today, in terms of modifying the whole red blood cell and making it a new home for the parasite. The parasites that are being emphasized in the MAPIC are falciparum, but as a model we use Plasmodium coatnei. Both of these parasites are very similar in their biology and pathogenesis. Both of them have these knobby protrusions we heard about today that are responsible for the cytoadhesion and sequestration of the parasites and all the associated pathology. We also then are working with vivax and its model Plasmodium cynomolgi. Now the surface structures for these parasites are very different, and the pathology as well, anticipated to be. This is what the parasite does at the surface of its red blood cell. It makes hundreds of these complexes, seen here for the first time in 3 dimensions. They're called caveolar vesicle complex structures. The blue is the red blood cell membrane. These are cavernous structures open to the circulation. Here you see the speckling you see in a Galinksi stain of Plasmodium vivax or cynomolgi. Each little dot is one caveolar vesicle complex. And you can see here by EM, they're actually open. You see an indentation of the infected red blood cell membrane. So there's this communication that we don't understand. A lot of the biology of these things, of the parasite and the host communicating, has yet to be learned. Also for Plasmodium vivax and cynomolgi, you have the new form called the hypnozoite in the liver. You have a primary schizant growing, releasing thousands of merozoites, but then you have some go dormant called hypnozoites in the liver. and then they'll relapse. Could be weeks, months, or a year into infection. You can have a relapsing infection and the person is sick again. How these activate is largely unknown, though we've known there's been hypnozoites for about 20 some years, but we haven't known anything about them until recently. We're starting to get a handle, and I'd just like to highlight this paper that just came out, actually 2 papers. John Barnwell and I co-authored a paper called Malaria Liver Parasites Awaken in Culture. It's actually a News and Views article in Nature Medicine that actually describes and summarized the original article, Dembele et al. From last month, February, Nature Medicine. What they showed using a Plasmodium cynomolgi model that you can actually now, for the first time, culture the parasites in the liver cells and actually see the dormant forms grow again. So we now have a culture system that this group in Europe has developed to see what is going on when the parasites become activated again from dormancy. The last parasite that we're working and emphasizing in MAPIC is Plasmodium knowlesi. Quite different from Vivax and Falciparum and a model for both in different respects. It's different because it's 24-hour life cycle and the others are longer, 48. It's also nice because we can study it in the natural host with a mild infection, the experimental host with a virulent infection, the rhesus monkey. It's in humans. We have samples predicted to come from human samples, human populations, and also it can be cultured and studied in culture, like Falciparum can as well. Now the surface of this one is completely different, and it has neither knobs or caveolo-vesicle complexes. It only has an occasional caveolae. And that begs the question for me, I think knowlesi is a great model for what's fundamentally important. It doesn't need the fancy stuff. All it needs is this. And it does it in 24 hours, the growth in the red blood cells. So what are the basic underlying biology that's necessary for survival? I think we may learn something from knowlesi. We also have shown just that knowlesi, in comparing the in-host dynamics and gene expression with the cultured in vitro, you have a different gene expression, clearly telling us the host matters. So the work we're doing in context of the host does make a difference rather than only working in cultures. And in fact, we've shown with this work, this is microarrays followed up with RNA-seq, that you, even with the VAR gene counterpart in knowlesi, you see a complete turning on of a lot of VAR genes, but specific proteins are expressed. So you really need to compare the host and the in vitro. In vitro, you get downregulation of a lot of genes, including the VAR genes. And we will have a lot to learn compared what's important about the host for the parasite survival. So here's MAPIC overview. We have a lot of infections going on in the animals and also samples coming from humans. Samples are processed in our malaria core, and then they are aliquoted out for different cores to do immune profiling, innate and adaptive immunity, functional genomics, proteomics, metabolomics, and lipidomics. We have a lot of in-house analysis in the different cores, and then data coming out to UGA for the informatics core, where it's processed further and made usable, both for the cores again to do further analysis together, and for the mathematical modelers in the final element here to actually start to build models and come and test different experimental scenarios in silico, and then help to guide our future experimentation. Don't expect you to read this, but just to also just to make the point, these are very dynamic plans. We have this all running now. The first year and a half, we've got the whole system running. We've done experiments with Plasmodium coatney for 100 days, or we're in process with that, I should say. Plasmodium cynomolgi already done for 100 days, controls for 100 days. And now we're kind of looking to be sure the biology really meets our needs as we continue future infections, to study recrudescence, to study relapse, to study co-infections, and to really understand the relationships of the monkey model malarias with human studies. Just an example of that 100-day infection. We start with sporozoites, not from the mosquito, but from a syringe, but that's still going to give us an idea of what's happening, and then we can monitor from the beginning innate immunity that's going on, parasitemias, watch the parasitemia come up, watch anemia set in, watch severe disease set in. So every day we're monitoring the clinical signs and the parasitemias. Every other day we're getting samples for metabolomics, and then at specific time points we're getting samples for all the other omics. This is modeled after another paper that we published recently, Moreno et al. In Infection and Immunity in 2013, specifically watching a Plasmodium knowles-i infection and how it does actually mimic a lot of the human severity. The modeling, I'm not a mathematician, but I'm guided by great mathematicians and systems biology experts who make it kind of easy to understand. It's just about taking data, ideas, drafting models, analyzing, more data comes in, refine, reality check. And when you have an expert like this publishing a systems biology book that's rather straightforward for students, highly recommended, he kind of makes it easy for us to understand how this process will work. And if you keep doing it, you get insights. And now, knowing Professor Eberhard Voigt from Georgia Tech for 3 years, we're starting to see this process in action. We are starting to get insights from data that they've been modeling in the mathematical modeling core. And one of the areas of most interest is, for example, anemia. So there's several things of interest: the whole parasitism of red cells, the anemia, the immune response, the hypnozoites, the relapses. There's lots of elements of, lots of parts of the malaria process of infection and sickness and immunity that we want to model, but this is one of the predominant ones. At year one, we've been working through a lot of processing of data. This is happening at UGA. Just moving secure data for all these institutions has been one of the major feats. Being able to have data generated, move through various servers, move off to UGA, process there in a way that also it can be pulled back by the different cores. And it's an interesting process. I'm not going to read all of this, but first and foremost, you want to see the data coming into the UGA, not going between cores. But then once it comes from UGA, we can all work with the data to see how it all is integrated. Ultimately, to be getting the information, as I said, to the community at large. And to acknowledge the full team at UGA, we've got, in addition to Jesse Kissinger, we've got Juan Gutierrez, who you'll be hearing from today, and also Jeremy DeBerry has been here in the back row with us today. The longer-term view is the database systems that are being developed to study MAPIC with the community at large can be also used and developed for other pathogens. And again, this is involving host and pathogen, quite unique from other database systems accessible to the malaria community so far, such as PlasmoDB, which is about the parasite. So this is going to be unique, parasite and host. In the modeling team at Georgia Tech, we also have Mark Styczynski. His main role is taking all this omic data as well as clinical data and creating pipelines for us to be able to access that information and understand that data. Big challenging to do one at a time, let alone to to understand how they are all related to each other. That is shown here in the next slide. The goal of all these different core leaders is to integrate the data. So how does the genome relate to metabolome, transcriptome, metabolome, proteome, metabolome, et cetera? And that's a lot of new things that we're going to have to learn to understand how these things relate. And it's another one of our major challenges. Innate immunity, followed by adaptive immunity. Of course, we're not going to go in detail. We have a deputy core leader here, Tracy Lamb, in the room for the innate core. But there's a lot of things that we simply don't know about innate immunity. Same thing for adaptive. A lot of things we just don't know. Malaria field has moved from understanding things in terms of ELISAs or ELISpots or different kinds of assays, but not on a grand scale like this. So the team working on immunology, as noted here, that's who Tracy is here with us today. We have just a nice group of experts who are doing a very intensive work to come up with different panels for intensive flow cytometry analysis predominantly and other types of assays to understand the immune response. Last but not least, metabolomics. We're using high-resolution metabolomics to actually understand the metabolites that one finds from host and parasite in very small samples of blood. And that's, again, the primary interest, but Dean Jones, leader of our core from Emory, Professor Jones, is interested in all the different aspects of the life cycle. And so we're, of course, looking to grow projects that would involve studying metabolomics of the different stages. And it's become so powerful. you can now measure over 20,000 different metabolites in very small biological samples. We're talking about 60 microliters of plasma, able to get this kind of data. And Dr. Jones is an expert in being able to analyze this data. His team is trying to also make it accessible to people who are not experts in metabolomics and biochemistry. So Karan Uppal is a leader in trying to come up with a flow at Emory that we're calling it user-friendly. Of course, you need a lot of dedication, but we have an infectious disease fellow now who got excited about this as a clinician, and he's starting to learn these ropes. So I would say it is user-friendly for someone who wants to be able to become involved at this level of understanding what those 20,000 metabolites or more are and how they relate to the infection. In closing, or close to closing here, we are not just working with the non-human primates, but the human samples are very important to the project. We've engaged a scientific consultation group coming from these various countries in order to work and collaborate with people in the field. We collaborate with them. They're also great advisors. Particularly samples have already come from Brazil, and soon to come, we hope in the next 1 to 2 years, from Colombia, Ghana, India, Malaysia, Papua New Guinea, Peru, and Thailand. Several African countries are also showing interest. Particularly, what's also very nice is we work closely with isomers. They're called the International Centers of Excellence for Malaria Research. Those countries where there are isomers are noted in red. And that's another NIH-funded project, so that communication has been important as well for the project. In summary, as our advisors say, MAPIC is a daunting, overwhelming, intimidating project, and it's also beyond my area of expertise, people have told me, but that's the case for everyone in the great team. There's so many different elements of science here. Nobody's an expert in everything, and maybe just a few areas, but somehow this project is moving quite smoothly, I would say. We're moving along, we have the common goal of understanding malaria better, different types of malaria infections and understanding it in depth, coming up with possible biomarkers that may be used as diagnostic tools, maybe targets of intervention in drugs, maybe new vaccine candidates. And closing words are, it's not just interdisciplinary or multidisciplinary. New word I learned recently is transdisciplinary. Just because, as I said, there's so much here. We're speaking not just about different scientific disciplines, but different languages to understand each other and to be able to communicate to develop this program and keep it on track. So I'd just like to thank a lot of people. A lot of people have been involved from Emory University at different levels. Because of this project, we now have over 12 people working on malaria at Emory. And that's simply because we've reached out to people and they've just shown interest, they've got excited, and now new projects are developing with those people from NIH grants, maybe a training grant, and so forth. So we welcome you if you have ideas from what you've heard today and want to reach out to discuss possible projects and lead them. We don't want to lead everything, we just want to lead this, what we're doing. We welcome the chance to assist in ideas that you may have scientifically. Again, Yerkes Primate Center, School of Public Health, Centers for Disease Control, Georgia Tech, University of Georgia, many international collaborators, and financial support from NIH. Thank you very much.
We have time for a few questions. Ah, come on.
Too daunting?
Comments, if not questions.
So Mary, I was going to ask, coming back, maybe something like what I alluded for diagnostic purposes. It will be really great to, identify. Really, there is a greater need, as you know. I don't have to convince you. We need. So it will be nice to identify some protein targets or any other targets, for that matter, that would be ideal candidates for next generation RETs. And we would be interested in exploring. Of course, John is right there, and it's also part of our interest.
Absolutely.
To talk more about it and see, like we have Kelly, like she's a bioinformatics fellow that CDC has come up with a new flagship program really to support bioinformatics applications. for public health needs. So we would like to really think about how we can make use of this rich data set that we're trying to.
Absolutely. We can be talking. Thank you. Yes,
Okay,
Naomi?
this is a basic question that I've been wondering for Vivax. I'm not very familiar with Vivax, but you do have. So what I know is that they infect reticulocytes, and that's one of the reasons why we can't culture them.
Mhm.
But I'm just starting to wonder, in real human infections, are they just infecting reticulocytes, or do they also affect mature RBCs?
You know, the word is reticulocyte specific and reticulocyte preference, and they may be, majority may be, in fact, the youngest red blood cells. But I bet there's a fine line there that gets crossed. In cynomolgi, for example, you can have quite high parasitemia. So clearly, the parasite sometimes may be more restricted, depending on the isolate, to reticulocytes, but then more mature cells can become infected. It won't surprise me if Vivax has something similar, depending on the isolate. I mean, and it's not many people study Vivax and invasion and those receptors just yet.
Eldon?
Are there plans to look also at the host side of things? Because I'm here focused on the parasite, but I feel like eventually they're going to ask to look at variations on the host side.
In fact, a requirement of the contract is parasite and host. So when we're talking about the omics, we are getting both. Transcriptomics, it's the host and the parasite. Metabolomics, host and parasite. Proteins, host, parasite. Immune profiling, it's about the host. So everything here is definitely host and parasite. And we have to think also in broader terms beyond malaria. So we think co-infections with schisto maybe someday, but we're getting the underlying work done in the animals with controls and malaria infections and maybe malaria co-infections that may lead to be able to do this kind of systems biology work maybe with other organisms as well. Yes?
Just going by the talk this morning about arthropods not being as dangerous, are you also looking at infection through mosquitoes? Is that part of it?
Yeah, only 19.4 million. We couldn't get, we had to convince reviewers as much as they thought too. The sporozoite side was interesting, the liver, the vector, we agree, gametocytes. But really, when you just want to get a hold of what's going on in the blood stage infection, that gets spread thin with a group like this doing what we're doing. But we are interested in the vector. And in fact, some of our immunologists, I don't know about Tracy, but I know Rabin Taravanzian is interested in the mosquito immune system. He says whenever we're getting sporozoites and we go for the salivary glands, he wants whatever's left. And so, there are those kind of potential even piggybacking onto what's already being done. So we hope to get more into vector. We do see gametocytes. We have a student who's moving into gametocyte work, a student who's become interested in microvesicles, and that's also might be relevant to the liver and might be relevant, in other aspects of the life cycle. So we're in the growing process, starting where we have, but please feel free to contact anybody on the team if you have ideas that can be piggybacked on this.